All right, so the next one we want to talk about is who actually came up with um, the structure of DNA, and that would be Watson and Crick. And James Watson is the one sitting down here, and Francis Crick is the guy standing up. And the two unknowns, they were working at the University of Cambridge, and they were kind of working together. Over the years, they had a falling out. They really didn't like each other all that much. But in 1962, they were awarded the Nobel Prize. So Watson and Crick are well known for this. But what I do want you to know is there was a third person that was actually also won it. Oh, that's not him. This is James Watson. Picture taken going on maybe about 10 years now. So he was also a professor at UCLA later in life. He was actually a retired professor. He actually had an office, or still has, maybe still even has an office at UCLA, and writes textbooks. And I laugh at that because at one time here on campus, we were using that textbook, and his name was first on there, but the authors were actually asked on how much he had actually written, and they said he's a big picture kind of guy, which basically means he kind of came up with some ideas on how it should be put together, but the nuts and bolts were pretty much written by everybody else. So, but anyway, so the other guy that won the Nobel Prize was Maurice Wilkins, and he was doing X-ray diffraction at King's College in London. He was actually friends with Watson, and so he was the third person to actually get the Nobel Prize. It turns out that at King's College in London, Maurice Wilkins actually worked with Rosalind Franklin. And this is the very famous picture. This is called Photo 51 of an X-ray diffraction picture of a DNA molecule looking straight down. The reason that this is such a famous picture is to kind of get you this. So these two guys are working on it. They are touring this lab. They knew about X-ray diffraction, and Maurice Wilkins actually was working on this, also taking pictures. And he had a grad student, and that grad student actually transferred to Rosalind Franklin. Now, these two did not get along. And so he was very quiet, very reserved, and being a female in science, at that time, Rosalind probably had to be a lot more forceful, and actually did not put up with a bunch of crap, which all the power to her. So unfortunately, though, so what happened was when they were touring, Wilkins actually took them into Rosalind's Franklin's office, and actually gave them a copy of that picture. And with that picture, they were able to win the Nobel Prize. He gets credit for the X-ray diffraction. These two get credit for coming up with the shape, and she gets absolutely nothing. So again, a bit rough for Rosalind. Now, there is another reason behind that, and it is because she had actually died before the gift was given. And so one of the things when you win a Nobel Prize, you get lots of money. Today, it's over $1 million. And you can do whatever you want with that money. But you have to be alive. Yeah, you can't win this posthumously. And she had died from cancer before the. So the thought process is she probably would have also won the Nobel Prize, along with the other three. Yeah, but the $1 million is great. Obviously today, I'm not quite sure how much the money was back then. Somewhat in the equivalent, I'm sure, in those days money. But even the bigger prize for a scientist winning the Nobel Prize is not just the $1 million, but it's also the prestige and the fact that the way we get money for research today is through grants and donations and things like that. You win the Nobel Prize, people throw money at you, and you're pretty much set for life as a scientist. So here's what I'm asking: if any of you get a Nobel Prize, what I want is not any of your money. So keep your money. All I want, I tell all my students, is I want a shout out. So when you accept the Nobel Prize at the award ceremony, I just want, hey, a shout out to Mark Cooper at Mount San Antonio College. That's all I'm asking. All right, let's take a look at why that picture was so important. One is it told them it was a double helix. And by looking straight down on it in that picture, what they did is they lined things up. And what they realized is that two purines next to each other were too wide, two pyrimidines together were too narrow, but a purine and a pyrimidine side by side fit perfectly into that picture. And then also with Chagos rule, guanine hooking with cytosine because there are 3 hydrogen bonds, and adenine hooking to thymine because there are 2 hydrogen bonds. That fit perfectly. So that's how they came up with the double helix. And that's pretty much where that aha moment came from. All right, so some basics I do want you to know. They do show up on most standard tests, so probably a good idea. First of all, I do want you to know that the base, okay, so the backbone of the sides or backbone of the DNA molecule is the phosphates and the sugars. Phosphate, sugar, phosphate, sugar. Okay, again, those are those phosphodiester linkages. So you need to know that. You also need to know then the rungs of the ladder, the steps would be the nitrogenous bases. And again, they're held together by hydrogen bonds. That's something to remember for later. The other thing you need to know is, and we'll talk about this, is you can see they've circled here, it's a 5' end and there's a 3' end, and we will talk about what that means a bit later. Okay, so the space between the two bases is 0.34 nanometers. One full turn every 10 bases. So this thing twists. And so there are 10 base pairs per twist. And for every twist is 3.4 nanometers. And then the distance from side to side of a DNA molecule is 2 nanometers. So I do want you to know those numbers. All right, and then here's a little more simplified. Again, the sugar phosphate backbone, they usually draw that in blue. And then the nitrogenous bases, or the rungs of the ladder, are the ones in the middle of the steps. The other thing I would like you to realize, and I'm not sure it's always real clear, is the nitrogenous bases are always attached to the actual pentose sugars. All right, so here's some very simplified pictures showing you some different looks on how they represent DNA structure. Notice again the 5' to 3', 3' to 5, 3' to 5'. So we will be talking about that on the next one. I guess I should mention it now. Let's go back here. If you look real closely here, you can see that there are these little tiny numbers in red here, or pink, I guess that is. 1, 2, 3, 4, 5. Organic chemists are anal. And so what they do is they actually number the carbons. And so the 5' end means it is the 5th carbon that is actually exposed. And if you look over here, this is the 3' end. It is the 3' carbon that is it is the 3rd carbon that is exposed on this side. That's going to become important when we replicate DNA. So you want to realize that. Okay, 5', 3'. All right, and then the last thing we're going to talk about is that DNA strands are not identical to each other. They are complementary. What that means is that you can have a strand of DNA on one side, and the other strand is not exactly the same as you can see here in B, but you can predict what it's going to be. Complementary means that, the A is always attached to T's, C's to G's. So they're complementary to each other. If we have one strand, we know what the other strand is going to say. All right, with that, we'll stop the share and stop the recording.